now we are going to the first part of this talk. And we are very happy to invite Fangyu Zhang, who comes from the University of California, San Diego. And he is working with Professor Joseph Wang. And his talk will be on ACE2 receptor-modified algae-based microrobots for removal of SARS-CoV-2 in wastewater. So without further ado, I just pass the stage to Fangyu. So please go ahead, Fangyu.
Yeah, thanks. Yeah, thanks for the kind introduction and the invitation. So my name is Fangyu Zhang from the Department of NanoEngineering, UCSD, and my advisor is Joseph Wang. So here I will share my presentation of the work which published in the JACS recently. So the title is ACE2 receptor-modified algae-based macrorobots for removal of SARS-CoV-2 in wastewater. So first, I will introduce some background about the SARS-CoV-2 with different possible transmission routes. So actually, the SARS-CoV-2 can spread through multiple transmission routes. For example, the close contact and between the person, also the direct airborne transmission from respiratory droplets or the aerosols, and also some indirect format transmission by the contacting the contaminated surfaces or the subjects. So it is already well known this SARS-CoV-2 can infect the respiratory tract, but it can also infect the GI tract, the gastrointestinal tract, with a prolonged residence in the fecal samples. So here it shows some simple ways for the fecal-oral transmission routes. So they can follow some paths such as the field, the flues, the flies, the fingers, and the formats. So in addition to the existence in the stool samples, so this SARS-CoV-2 has also been detected in domestic wastewater, in sewage, and downstream rivers. So actually, this entry of the virus into the sewage systems can lead to many potential transport pathways. So, and in the industrialized countries, most of the collected domestic wastewater and the viral load is treated in centralized wastewater treatment plants, also called WWTPS. However, this conventional WWTPS do not remove the virus completely. So these high infant viral loads during the pandemic can lead to the insufficient reduction of virus before discharge. So furthermore, the lack of the fresh water can also cause a reuse of the treated wastewater for many purposes, such as the groundwater discharge, groundwater recharge, the recreation and irrigation of the food crops. All this process will create other potential routes for SARS-CoV-2 transmission. So it is critical to evaluate the potential of the wastewater as a transmission pathway of SARS-CoV-2. So to minimize the risk of this secondary transmission to humans by the wastewater, so it is urgent to develop wastewater treatment strategies for effective removal of SARS-CoV-2. So here shows the typical wastewater treatment strategies. So the wastewater first undergoes the primary treatment, the virus removed by the sedimentation, but this efficiency is very low. And the secondary treatment using the biological treatment can combine the aeration tanks with some secondary sedimentation to retain the activated sludge. So this virus assumption to the organic particulates and removal by settling play an essential role in these secondary treatment steps. And this secondary effluent can be directly discharged to the surface waters without any other treatment or disinfected prior to the discharge. It can be further treated with advanced oxidation process called AOP, and also the filtration, where different membrane systems such as the ultrafiltration for the reuse applications. Although this conventional wastewater purification approach are effective in general, it is still necessary to explore some new techniques, which need to be easy, fast, and effective in resolving the wastewater contamination of SARS-CoV-2. So here we turn to the macromotor or the nanomotor for the specific environmental applications. So we all know the macro or nanomotor act as an artificial active matter, and they can enhance the reaction yields and reduce the treatment times by improving the interactions between their active surface and target pollutants. For example, the first example shows the Sanchez group used the graphene oxide-based Microrobot as an active self-propelled system for the capture, transfer, and removal of the heavy metal. So this active motion of graphene oxide platinum motors improve the mixing and mass transfer, and also enhance the rate of various remediation process. So the second example is from our group. We fabricated a light-activated titanium dioxide magnesium macrospheres. And these macrospheres can propel autonomously in natural water and for some photocatalytic degradation of chemical and biological warfare agents. So in such case of the bubble propulsion mechanism, so the bubbles can create additional mixing. So this can increase the CBWA removal efficiency. So the third example is from Qianghe's group. So they made a genus gallium zinc macromotor with good biocompatibility and biodegradability for active target treatment of the bacterial infection. So although such synthetic macromotor provides efficient and rapid decomposition of and accelerate the removal of pollutants, but the widespread environmental applications of the current synthetic macromotor platform still meet several obstacles. For example, the short lifespan using the magnesium motors and the need for toxic fuels with the hydrogen peroxide, and also the complex external actuation equipment using the magnetic propulsion equipment, and also the restricted operation medium for using the acoustic propelled mechanism. To address these challenges faced by synthetic macromotors, the biohybrid macromotors, combining the self-propelled microorganisms with some functional biomaterials, show some great promise for large-scale environmental remediations. So here we select the green macroalgae called Chlamydomonas reinhardti as a model algae for our research. So such algae have many attractive properties. For example, they can be reproduced in a large scale and also have a fast motion in diverse aqueous environments. And also they have a long lifespan in this environment. Also the surface can be easily functionalized with different chemical groups for further applications. So here we are thinking, can we use this algae for the SARS-CoV-2 removal? So in order to achieve these purpose, we need to functionalize the algae by some materials. So here we go back to the SARS-CoV-2 binding mechanism and infection mechanism to human. So we can see here the SARS-CoV-2 is covered by the spike protein, and they contain a variable receptor binding domains called RBD. And this RBD can bind to the angiotensin converting enzyme 2 called ACE2 receptors in the host cells, for example, located in the lungs, heart, kidneys, and GI tract. And actually, this binding between the RBD and ACE2 is very tight. So we are thinking, can we use this ACE2 receptor to combine with the algae for further removal of the SARS-CoV-2 spike protein? So here is our design. We propose an idea to combine this algae with the ACE2 receptor. So the ACE2 robot, algae robot is fabricated using a click chemistry for anchoring the ACE2 receptor onto the surface of the algae. Specifically, this azide can be functionalized onto the ACE2 receptor. So this azide can be functionalized onto the ACE2 receptor, and this DBCO can be functionalized onto the algae surface. This method can be used a amine NHS ester reaction. And after this conjugation, the azide and the DBCO can be conjugated together using the click chemistry. And thus it can form the ACE2 receptor-modified algae. And this ACE2 receptor would be responsible for the recognition of the target as protein or the virus with a high binding affinity to this S1 subunit of the virus spike protein and the SARS-CoV-2. The next, we confirm this successful DBCO NHS ester conjugation onto the algae. We are using some fluorophore labeled azide to confirm. So after this FAIM azide conjugation, so the algae show a fluorescent signal. So from the fluorescent images, we can see there is a colocalization of the algae itself and the fluorescence, which is coming from the FM azide. And next, we performed to visualize this ACE2 receptor attached onto the algae surface by some immunostaining. So here we use a fluorophore 488 conjugated anti-ACE2 antibody to label this ACE2 receptor on the algae. So such antibody can specifically label the ACE2 receptor and show the green fluorescence. We can also see this green fluorescence from the GLP channel can colocalize those of the autofluorescence of the algae in the Cy5 channel. In addition, the fluorescent intensity data and the flow cytometry histograms also confirm the ACE2 receptor binding. So from the chart, we can see the unmodified algae incubated with this fluorophore label antibody show a negligible change of the GLP fluorescence compared to this bare algae. In contrast, these ACE2 algae robots incubated with the fluorophore label antibody show a significant increase in the fluorescent intensity compared to these two control groups. So such results further reflect the effective conjugation of the ACE2 receptors onto the algae by click chemistry. Next, we investigate the motion behavior of such biohybrid motors. So the speed of the DBCO modified algae and the ACE2 algae robots was similar compared to the speed of the bare algae. So such results also indicate the fabrication process is biocompatible and has a negligible effect on the motion of the algae. And next, we continue to test the movement of such robots in various aqueous medium. And we can find that the robot can move in an effective way. The speed is more than 100 macromotor per second. So, and without any external fuels. In addition, such ACE2 algae robots demonstrate a very long-lasting motion in both drinking water and river water. So this data also displaying this ACE2 modified algae have capability of extended operation toward the removal of SARS-CoV-2 protein and S protein and the pseudovirus. So in the next part, I will focus on the removal of SARS-CoV-2 spike protein by these ACE2 algae robots. So in this figure, we can see the presence of the S protein in the drinking water showing red and the river water showing blue doesn't affect the mobility and the lifespan of the robots. So this means the robot can operate in such environments for a very long time scale. So next, we study the effect of the ACE2 robot density on the kinetic removal efficiency of the S protein from the drinking water sample. So here we can see it can remove more than 95% of the S protein by using a 5 × 10⁷ Lg per milliliter. And this process is just using a 6 hour. So by using the same density of the algae, we also compare the spike protein removal with different treatment groups. So we can see the robot group show a highly efficient binding and more than 90% removal efficiency. And in comparison, the active barrier algae without ACE2 receptor and the static algae without motion ability just display 45% and around 20% removal efficiency after a 16-hour operation. And the ACE2 algae robot also show an effective S protein removal in different medium. For example, the DI water, drinking water, and river water. The next part, after confirming the S protein removal by robots, we move on to the virus removal studies. So here we use a baculovirus presenting some SARS-CoV-2 spike protein on the surface as the pseudovirus. Before testing the virus removal, we double-check the motion behavior from both speed and lifespan of these robots in various water matrix containing the SARS-CoV-2 pseudovirus. It also shows a good motion ability and good lifespan in these mediums. So after the motion in the virus contained environment, we found there is a significant change of the ACE2 algae robot surface morphology. So we can see from the right SEM image, it shows the robot can bind to the virus after motion in virus water matrix. And you can see it has a relative rough surface morphology compared with the left one, which just move in the water without virus. So this data also verify our system has a great capability to remove virus in the water matrix. So next, we quantitate this virus removal efficiency. Here shows several steps for the quantification. So the after ACE2 algae robot treatment, the pseudovirus can be largely captured and removed from the solution. So it can be validated and visualized by an NL20 cell-based assay. So briefly, these NL20 cells remain non-fluorescent following 24-hour incubation in this ACE2 algae treated virus sample. But in comparison, so without this ACE2 algae robot treatment, the cells will display the bright fluorescence. So it reflects the pseudovirus entry and significant expression of the fluorescent protein in these host NL20 cells. So as follow the S protein removal studies, we also investigate the density effect on the removal efficiency. So from the results, we can see this highest robot concentration result in a minimal fluorescent virus signal. So which means it has a higher virus removal efficiency. And also this ACE2 algae offer the significant improvement in the removal of S protein, SARS-CoV-2 pseudovirus compared with the control groups, including the ACE2 algae, static ACE2 algae, the bioalgae without ACE2 receptor functionalization, and also the cell wall deficient algae. So we can see the robot after robot treatment, so the virus can be removed by more than 90% compared with the other three groups just has around from 20% to 60%. So furthermore, we also evaluate and compare the virus removal kinetic profiles in the drinking water treated with this ACE2 algae robot, also the bare algae, and also the static ACE2 algae. So such results indicate the significant contribution of the both ACE2 receptors for virus targeting and the fast macromotor motion for contacting the virus and towards the highly efficient viral removal and removal. And the last part, a post-treatment of the ACE2 algae robot by flocculant was performed to clean this robot sample in the water. So here the flocculant can isolate the ACE2 algae robot from the water matrix. And you can see that such process will not affect the virus removal efficiency. And so that means it will hold a considerable potential for future practical applications. So here is the conclusion. So in this study, we fabricate the ACE2 algae robots by an efficient click entry approach. And this approach doesn't hurt the motion behavior of the algae and the function of the ACE2 receptors. Also, by developing this biohybrid macromotor, we can achieve an effectively removal of the SARS-CoV-2 spike protein and pseudovirus from the various water matrix. So I will thankful to my advisor, Professor Joseph Wang, and our collaborator, Professor Fangyu Zhang, and all the group members from both groups. Yeah, thank you all.
Okay, thank you, Fangyu. Thank you for your nice talk. And now we're going to the Q&A session. And if audience have questions,